Hello everyone, welcome to our second installment of our Microbiome Foundations webinar series. I'm Dr. Sarah Ellis, and I'm so excited to chat with you today about the systemic impact of gut dysbiosis. Now we have quite a bit of ground to cover today, so let's jump right on in. During our time together today, we'll start with a brief overview of gut dysbiosis. We'll discuss the differences between a healthy and a leaky gut, and then we'll get into the details of the impact of gut dysbiosis on our overall health. We'll cover the gut-brain axis, gut-heart, gut-lung, gut endocrine, gut immune, and then we'll end with the gut genitourinary axis. Let's jump right on in. Let's start by discussing gut dysbiosis. Gut dysbiosis can be defined as a reduction in microbial diversity and is often a combination of the loss of beneficial bacteria and a rise in pathogenic bacteria. When we look at this image over here on the right, we can see that homeostasis shows a great blend of all of these different types of bacteria. They're existing in balance with one another. But dysbiosis can look like the expansion of and the overabundance of pathogens, as we see over here on the left. In the middle, dysbiosis can also look like reduced diversity. So the pathogens may not necessarily be overabundant, but there just is not that many different types of bacteria present in the gut. And then over here on the right, dysbiosis can also look like a loss of beneficial microbes. So again, the pathogens have not begun to take over the microbiome, but this patient is missing some of the key protective strains and beneficial strains from their microbiome. If you're looking on a stool test, there are a couple of signs that you can look for that are key indicators of gut dysbiosis. For example, if you're looking at our BiomeFX stool test, you may see a decrease in alpha and beta diversity, so a decrease in both the number and quality of species in the microbiome. You may also see decreased protective keystone species, a decrease in the production of beneficial byproducts like vitamins and short-chain fatty acids. And you may also see an increase in the presence of pathogens and an increase in the production of harmful byproducts like histamine, phenols, polyamines, and many others. And then, of course, in general, you may also see an increase in inflammatory markers as well. While antibiotics are certainly a risk factor for developing dysbiosis and leaky gut, it's important to note that there are a variety of additional risk factors that can compound the problem. They include natural antimicrobials, pesticides, infections, excessive alcohol intake, smoking, stress, lack of sleep, intense exercise, and a poor diet that usually looks like the standard American diet or one that is high in saturated fats. As a functional medicine practitioner, it is so important to consider the variety of risk factors that our patients are exposed to every day and that further compounds their risk of developing gut dysbiosis. So let's start by taking a look at the differences between what a healthy microbiome looks like and what a dysbiotic microbiome looks like. Let's start on the left. A healthy microbiome will be one with low levels of pathogens, a strong mucosal barrier, low mucosal inflammation, and strong tight junctions between these colonocytes. Now, a dysbiotic gut will start with changes in the diversity. So we'll begin to see an overabundance of mucus degrading and toxin producing pathogens. We'll begin to see specific toxins and endotoxins like lipopolysaccharide or LPS. We'll also start to see some of these tight junctions begin to weaken. And of course, as those toxins begin to pull through those loose and weakened tight junctions, we'll begin to see inflamed intestinal cells. As those toxins begin to pour through into circulation, our macrophages and other immune cells will begin to pick them up and will activate the inflammatory cascade, which usually looks like a increase in the presence of certain inflammatory cytokines like interleukin 6, TNF alpha, and interleukin 1 beta. These dysbiosis risk factors, especially antibiotics, can change who is in the gut by eliminating or suppressing commensals. Now, this allows room for more opportunistic and pathogenic bacteria to run rampant. Below, we will see screenshots from our BiomeFX microbiome analysis. Alpha diversity is going to refer to how many species are in the gut. High diversity is usually associated with a healthy microbiome, as the competition between species ensures that no one microbe overgrows and creates an infection. Now, dysbiosis risk factors like antibiotics and other factors like stress and a poor diet will decrease overall diversity, whereas beta diversity will refer to the quality of the bacteria. Here on this sample, we see that this patient has 98 species in their gut microbiome. And on first glance, we may think that, wow, that's almost 100 species. That's great. But really, in a healthy human gut, we would like to see closer to 200 different species in the microbiome. We also want to take a look at the core microbiome. So the 4 major groups of bacteria that's present in the microbiome: Bacteroidetes, Firmicutes, Proteobacteria, and Actinobacteria. When we begin to see an imbalance between these 4 species, that is also a sign of gut dysbiosis. And then, of course, on our test, we also look at the percentiles and how well balanced these groups of bacteria are. If you find that your patient has an imbalance and has an overgrowth of Bacteroidetes, for example, and then not enough Firmicutes, that is also a sign of dysbiosis. Again, these dysbiosis risk factors can change who is in the gut. And when the who changes, the what is also going to change. Different species of bacteria carry out different functions and can either be helpful or harmful for us. For example, commensal bacteria are known to produce health-promoting short-chain fatty acids like butyrate and acetate and propionate. They also produce vitamins, antioxidants, neurotransmitters, and can help optimize the gut pH, which supports the growth of healthy commensals and can discourage the growth of opportunistic and pathogenic bacteria. But on the other hand, those pathogenic bacteria, instead, they can produce more harmful byproducts like ammonia, hydrogen sulfide gas, methane gas, toxins and virulence factors, and particularly a very common endotoxin called lipopolysaccharide. The focus of our time together today will talk about gut dysbiosis and its overall impact. Clinical research has shown that the microbiome plays a role in metabolic dysfunction, autoimmune disease, skin conditions, particularly eczema, nervous system disorders, hormone balances, liver health and function, and mood disorders like anxiety and depression. As we can see from this image, dysbiosis of the human microbiota affects nearly every organ in our body. Dysbiosis can affect our metabolism and can lead to obesity, type 2 diabetes, and mitochondrial dysfunction. Dysbiosis across the gut-lung axis can play a role in the development of asthma, obstructive pulmonary diseases, and even lung cancer. The gut-brain axis has been well studied, and there have been numerous studies showing the connection between gut dysbiosis and psychiatric diseases, autism, and even neurodegenerative diseases like Alzheimer's and Parkinson's. There's also a gut-heart connection, and later on today we'll talk about how gut dysbiosis is connected to atherosclerosis, thrombosis, and even heart failure. And then, of course, gut dysbiosis is connected to the skin as well, particularly as it pertains to psoriasis, acne, and even potentially skin cancer. All right, let the fun begin. We've had a great time setting up a foundation, discussing the differences between a healthy gut and a leaky gut, and what exactly dysbiosis means. Now let's walk through the different gut organ axes and discuss the ways in which dysbiosis can impact those different organ systems. We are going to start from the top and work our way down the human body. So let's start by discussing dysbiosis and the gut-brain axis. The one fascinating thing about the gut is that it is powered by its own nervous system, the enteric nervous system. Now, there are a lot of neurons in the enteric nervous system. In fact, there are more neurons in the gut, over 100 million, than there are in the entire spinal cord. So today we are going to talk more about the main neurological system that the gut is exposed to, particularly the impact of the vagus nerve as a connection point and communication pathway between the gut and the brain. It's important to note that the enteric nervous system is fully accessible to the microbiome and operates independently of the central nervous system and peripheral nervous systems. The gut-brain axis is a bidirectional communication pathway between the gut and the brain. The primary mode of communication between these two organs is the vagus nerve, which is our longest nerve and our 10th cranial nerve. The gut-brain axis regulates GI homeostasis. It connects emotional and cognitive areas of the brain with specific gut functions. And this gut-brain axis involves many different organ systems. It involves the nervous system, of course, which includes the vagus nerve, the brain, and the spinal cord. It also involves the endocrine system, specifically the HPA axis, the immune system as well, and of course, the gut microbiome. As I mentioned in the previous slide, the enteric nervous system contains more nerves than the spine and produces more than 30 different neurotransmitters, all of which have an impact on brain function and gut function as well. What's really interesting is many of the hormones and peptides that are produced and released by the enteric nervous system can cross the blood-brain barrier. One peptide and hormone that we think about is ghrelin, our hunger hormone, that is able to cross the blood-brain barrier and can change our hunger cues. When we think of the communication style between the gut and the brain, there is a difference in which organ is sort of the chatty or extroverted friend and which one is more quiet and introverted. If you had to guess, I'm sure you'd probably say that the gut is the extroverted friend in this dynamic duo, and you would be correct. In fact, researchers have found that 90% of the signals from the gut, between the gut and the brain, come from the gut communicating to the brain, telling it that we're hungry or that we're full, or these are sort of the compounds and molecules that the microbiome has been able to make. And instead, the brain is more of the quieter, introverted part of this duo, whereas 10% of the signals come from the brain directly to the gut. As I mentioned before, the vagus nerve is the 10th cranial nerve and is the fastest and most direct route that connects the gut and the brain. The name vagus nerve is derived from Latin for wandering, and that's due to its extensive innervation. We can see here that there are so many neuronal projections off of the vagus nerve, and that is because it collects all of the information from many different visceral organs and shares it with the brain. It innervates the pharynx and larynx in the neck that allows for swallowing. It provides parasympathetic supply to the heart to help reduce our heart rate, In the intestines, the vagus nerve helps to regulate contraction of the smooth muscles, participates in GI motility, and even is involved with glandular secretions. Also, it has different receptors in the esophagus, stomach, and even the proximal small intestine. So the vagus nerve is quite involved in GI function. It's also responsible for regulating internal organ function, so that's digestion, heart rate, and even our respiratory rate. It is also involved in vasomotor activity and even certain reflex actions. So we're talking about coughing, sneezing, swallowing, and even vomiting. I've taken the time to highlight the importance of this bidirectional communication and the impact that the vagus nerve has because of the rise of functional GI disorders. Now, these are disorders that are defined as a group of syndromes that are characterized by a specific blend of chronic GI symptoms. These symptoms usually include abdominal pain, dysphagia, dyspepsia, diarrhea, constipation, and bloating. You'll see here over on the right that there are, according to the Rome 4 classification system, there are 33 adult disorders and 20 pediatric functional GI disorders. They range from irritable bowel syndrome, functional heartburn and dyspepsia, and even functional constipation or diarrhea. Now, all of this is really interesting because these conditions arise due to alterations in gut-brain communication. In fact, 2/3 of patients with functional GI disorders also have psychopathology, including anxiety and depression. In addition, more than 2/3 of patients with functional GI disorders have seen a doctor in the last 12 months, and 40% will use regular medication to help manage their symptoms. This presents a huge opportunity for integrative and functional medicine practitioners to provide support for patients who are experiencing and struggling with functional GI disorders. And one of the resources you can provide for these patients is supporting their gut-brain axis and reducing their gut dysbiosis. Now, of course, we have to have a pop quiz. So get ready to answer this question in the chat. Which of the following is considered a cause of gut-brain axis dysfunction? A, LPS endotoxemia. B, fasting. C, candida infections. Or D, none of the above. I'll give you a few moments to put your letter in the chat, and then we'll share what the answer is. I wish I had some Jeopardy music right about now. Great, I see some answers coming in. Wonderful. All right, the answer is A. Lipopolysaccharide endotoxemia is a common cause of gut-brain axis dysfunction. And so the next few slides, we're going to talk about exactly how that happens and how LPS can impact the gut-brain axis. All right, let's chat about lipopolysaccharide endotoxemia, a common cause of gut-brain axis dysfunction. Lipopolysaccharide is an endotoxin produced by gram-negative bacteria. Gram-negative bacteria actually make up 50 to 60% of the bacteria in our microbiome. So that means that over half of the bacteria in our gut contain this endotoxin. Now, I want to quell any concern or worry. When LPS is inside of the bacteria, it does not cause us any harm. But the problem is when the bacteria dies, they release LPS into the lumen of the colon. Now, if this patient has leaky gut and weakened tight junctions, the LPS can seep through those weakened tight junctions and enter circulation. Now, one of the first places that it will go to is the brain. And when it reaches the brain, it can wreak havoc. This study was published almost 15 years ago, and even back then, researchers were identifying the connection between the gut and the brain, and particularly the impact that the translocation of lipopolysaccharide, the impact that could have on mood disorders like depression. This study found that LPS from gut bacteria caused an inflammatory response that plays a role in the pathophysiology of major depressive disorder. Participants with major depressive disorder tend to have elevated levels of pro-inflammatory cytokines. They also tend to have poor cellular immunity, which kind of looks like low levels of natural killer cells, and they even tend to have tryptophan depletion. What researchers have found is that systemic lipopolysaccharide also increases inflammatory cytokines systemically and causes chronic central neuroinflammation. So essentially, that toxin that seeps out of the colon and enters circulation, once it enters systemic circulation, it can cause not just overall inflammation, but chronic neuroinflammation that impacts our brain. In animal models, the combination of central neuroinflammation and increased cytokines led to certain sickness behaviors that mimic depression. So those animals presented with anorexia, malaise, loss of interest, and other behaviors that are commonly seen in patients with major depressive disorder. Microbial mapping of the stool of patients with anxiety and depression found that they produced fewer short-chain fatty acids, so they had fewer bacteria like Faecalibacterium that produced butyrate, acetate, and propionate. They also saw that they had increased presence of bacteria that are associated with inflammation. So they had an increased presence of families like Enterobacteriaceae and Desulfovibrionaceae, two families of bacteria that contain pathogens that are known to induce gut inflammation. Not only is LPS implicated in the development and severity of mood disorders like depression and anxiety, but many researchers have found that lipopolysaccharide also plays a role in Alzheimer's and other neurodegenerative diseases. This study found that gut-derived LPS has a unique affinity for the nucleus of Alzheimer's disease brain cells. LPS was found to bind to the nucleus and actually prevent mRNA communication in the Alzheimer's brain. And the presence of LPS and its inflammatory activation is thought to be one of the main drivers of the development of amyloid beta plaques in the brain. This 2019 study also found a connection between the gut microbiome, intestinal pathology, and Parkinson's disease. This study found that the inflammatory response actually increases gut permeability and exposure to endotoxins like lipopolysaccharide. The exposure of these endotoxins and other bacterial products can actually induce alpha-synuclein aggregations, and that in turn allows it to propagate to the brain and the central nervous system via the vagus nerve. So again, the inflammatory process that begins in the gut does not just stay in the gut. It is able to, those byproducts from the gut microbiome and from those pathogens are able to travel upwards through and via the vagus nerve to impact our central nervous system and drive the development of neurodegenerative diseases. Lipopolysaccharide can also impact the way that our brain uses tryptophan. An altered tryptophan metabolism has been associated with a number of neurocognitive symptoms, including poor memory, poor sleep, and poor resilience to stress. Let's walk through the ways in which LPS modulates and drives this phenomenon. Let's start with the beginning. So lipopolysaccharide, as I mentioned, is a pro-inflammatory endotoxin and is a component of the outer surface of gram-negative bacteria. If a patient has leaky gut, that LPS molecule can translocate across the weakened tight junctions and across the colonocytes and enter circulation. Once it's in circulation, this is where it will activate the immune cells and the inflammatory cascade will begin. This usually presents as the production of pro-inflammatory cytokines like interferon gamma, TNF alpha, and interleukin 6. The presence of lipopolysaccharide also increases the enzyme IDO. IDO is an enzyme that takes tryptophan and turns it into toxic byproducts like kynurenine and quinolinic acid. This means that fewer amounts of tryptophan will be available to produce serotonin and melatonin for a balanced mood and better sleep. Now, this enzyme, like I said, turns tryptophan into toxic metabolites. And this reduces the availability of tryptophan to produce other important hormones and neurotransmitters. Inflammation can then determine the pathway of tryptophan metabolism, and determine whether it will go down the neurotoxic pathway over here on the right, or the neuroprotective pathway over here on the left. So let's walk ourselves through this schematic. So tryptophan can either be obtained through diet, or some of our bacteria can produce tryptophan. In a healthy person with a healthy gut microbiome, tryptophan will be used to make serotonin for a balanced mood and melatonin for improved sleep. When someone has sufficient levels of tryptophan and is shuttling it down this neuroprotective pathway, they will have restful sleep, a balanced circadian rhythm, a positive outlook and resiliency to stress, and a good memory. However, the presence of inflammation and even stress and excess cortisol can turn tryptophan into kynurenine and drive it down this neurotoxic pathway. Now again, lipopolysaccharide drives inflammation and even increases this enzyme here to turn tryptophan into kynurenine. So as we have increased inflammation, most of the tryptophan will begin to go down this neurotoxic pathway. And as this pathway becomes preferential in this inflammatory environment, patients will tend to present with a depressed mood, more irritability, a decrease in resistance to stress, poor sleep, and poor concentration. All right, we are making our way through the body. We just got done discussing the gut-brain axis and the way that dysbiosis can impact the development of neurocognitive diseases and mood disorders as well. So now let's talk about the gut-heart axis and the connection between the gut microbiome and our heart health, and even the development of certain heart diseases as well. Dysbiosis plays a critical role in the development of chronic disease, and that also includes cardiovascular disease. There have been numerous studies that have highlighted the crosstalk between nutrition, the gut microbiome, intestinal permeability, and the immune responses that affect cardiac homeostasis or even promote cardiac disease. Now, there are several microbial metabolites that have been identified as impacting the development of cardiovascular disease. These metabolites include TMA or trimethylamine, LPS, which we've talked about a lot so far, bile acids, serotonin, and even short-chain fatty acids like butyrate, acetate, and propionate. Let's talk about one of those microbial byproducts that we discussed in the last slide, and we'll talk about TMAO, trimethylamine N oxide. This is a compound that is produced by the gut microbiome from lecithin, choline, and L-carnitine-rich foods. It's commonly found in red meat and full-fat dairy, eggs, and fish. TMAO is produced when the TMA is metabolized in the liver. Now, TMAO is actually the biomarker that is known for its pro-atherogenic effects. So it's important to note that it is not the TMA that is produced by the bacteria in the gut, but the metabolism of TMA to TMAO that is known to cause a variety of issues with metabolic health. As I said, it's known for its pro-atherogenic effects and is positively associated with early atherosclerosis. It's also been associated with inflammation, poor cholesterol metabolism, and even thrombosis. A meta-analysis of 19 studies showed higher plasma TMAO levels have been linked to an increased risk of cardiovascular events like stroke, MI or heart attack, and death. In fact, a higher microbial diversity, so more bacteria in the gut and an improved diversity of bacteria in the gut, has been shown to decrease TMAO. Now let's take some time to discuss the impact that dysbiosis has in cardiovascular disease. A 2017 study analyzed stool samples from 218 people with atherosclerotic cardiovascular disease, or ACVD. They also analyzed stool samples from 187 healthy controls. The gut microbiome of these patients, those with the atherosclerotic cardiovascular disease, showed a number of patterns. What they found is that those patients had an overabundance of inflammatory bacteria, so the Enterobacteriaceae, E. coli and Klebsiella, and quite a bit of the Streptococcus species as well. This was coupled with a depletion of butyrate-producing bacteria like Faecalibacterium prausnitzii, one of our primary keystone species in the microbiome. We also saw a presence of inflammatory metabolites and toxins like TMA and TMAO, hydrogen sulfide gas, histamine, and LPS. This was also coupled with reduced fermentation of dietary fiber, which means a reduction in short-chain fatty acid production. We also noticed that bacterial DNA had been found in the atherosclerotic plaques, which highlights the affinity that lipopolysaccharide has not just for the amyloid beta plaques in the brain, in the Alzheimer's brain, but also the atherosclerotic plaques in our vasculature that plays a significant role in the development of cardiovascular disease. This table highlights the different types of cardiovascular diseases, the changes in gut microbiota that we see in these disease presentations, and also the involvement of different gut microbiota metabolites. So let's start at the top with coronary atherosclerosis. Researchers found that patients with coronary atherosclerosis tend to have increased streptococcus levels, increased ruminococcus levels, and increased Clostridium in their stool sample. The primary gut microbiota metabolite involved in the development of coronary atherosclerosis is TMAO. And this happens by reducing cholesterol metabolism, as well as promoting inflammatory transcription factors and inflammatory cytokines like NF-kappa beta, interleukin 18, and interleukin 1 beta. In hypertension, we saw an increase in Prevotella species, as well as increased Bifidobacterium and Lactobacillus. Now, what's interesting is these three groups of bacteria will all make short-chain fatty acids, but their impact is quite different on the body and on the heart. Particularly with the Bifidobacterium and Lactobacillus, especially the lactobacillus, they make short-chain fatty acids like propionate. And propionate can adjust the levels of T helper 17 cells and subsequently lower blood pressure. We can also see similar changes with heart failure patients, a decrease in the protective keystone species Faecalibacterium, which leads to a reduction in the short-chain fatty acid butyrate production, which is so important for healthy colonocytes and a healthy intestinal barrier. Dysbiosis also plays a role in coronary artery disease. A 2019 clinical study showed that gut microbial composition and metabolites changed significantly with coronary artery disease severity. In fact, the gut microbiome of patients with coronary artery disease showed the following patterns. They showed an increase in the abundance of inflammatory bacteria that belong to the proteobacteria phyla. Now, this is important because proteobacteria contain lipopolysaccharide, or LPS. The gut microbiome of patients with coronary artery disease also showed an increase in production of toxic metabolites like hydrogen sulfide gas and histamine, and also a decrease in short-chain fatty acid production, particularly a reduction in butyrate. A reduction in butyrate production in the colon can lead to increased gut permeability, which increases LPS translocation into circulation and an increase in inflammation. Dysbiosis has also been seen in hypertension. So hypertension is the most common risk factor associated with cardiovascular disease. The gut microbiome has been shown to play a role in blood pressure regulation, and when we compared these stool samples of those with hypertension to healthy individuals, we found that patients with hypertension tend to have a microbiome with low diversity, so they have low alpha diversity, which refers to the quantity of bacteria in the gut. And they also have low beta diversity, which refers to the quality of species in the gut. They tended to have an increased abundance in the Prevotella genus, as well as imbalances in protective bacteria like Bifidobacterium and Lactobacillus, as well as usually an increase in inflammatory bacteria like Streptococcus and E. coli. Now, these groups of bacteria are quite important because they can make neurotransmitters that can impact the autonomic nervous system, vascular tone, and ultimately blood pressure. As we've discussed earlier, TMAO is one of the toxic metabolites that can be produced in a dysbiotic gut, and TMAO has been positively associated with hypertension. Patients with hypertension also had reduced short-chain fatty acid production as well. What was most interesting is there was a study that conducted a fecal microbiota transplantation from hypertensive patients into germ-free mice. And what they found is that those germ-free mice, once they received that fecal transplantation from hypertensive patients, their blood pressure began to increase. So this tied together the connection between the gut microbiome and the heart, specifically the way in which our body is able to maintain vascular tone and blood pressure. All right, let's keep this party going and discuss the connection between gut dysbiosis and the lungs. Let's begin by discussing the connection between gut dysbiosis and asthma. Asthma is a common chronic respiratory disease that affects more than 300 million people worldwide. In fact, microbial dysbiosis within the lungs and gut has been associated with asthma. Now, asthma can be caused by many factors related to lifestyle and even environmental exposures. But many studies have underscored the importance of how a healthy microbiome in the lungs can play a role in the development, prognosis, and severity of asthma. A healthy lung microbiome is rather transient at steady state. Now, in contrast, during a disease process, the inflammatory conditions can enable the growth of certain bacteria that can lead to lung dysbiosis and subsequent colonization with pathogens. The concept of the gut-lung connection was born out of the observation that different lung diseases can be influenced by the intestinal microenvironment and vice versa. The gut microbiota of patients with asthma and severe bacterial pneumonia often differs from that of healthy controls, which we would obviously expect, as those disease states tend to be associated with an overabundance of pathogenic bacteria. But the effects of the gut microbiota on asthma are at least partially mediated by bacterial metabolites, particularly lipopolysaccharide. Now, the most known metabolite that is demonstrated to actually have protective properties in the human airway are short-chain fatty acids. Children with high amounts of butyrate and propionate in their feces at 1 year of age have significantly less atopic sensitization and are less likely to have asthma between 3 and 6 years of age. This is a schematic representation of different asthma phenotypes and endotypes partnered with differences in their microbiota. The endotypes classify asthma based on pathophysiological mechanisms, whereas the phenotype refers to the clinical and morphological description of the disease. So let's take a look at the two endotypes. Type 2 asthma, what we saw in the lung microbiome is that we saw an increase in inflammatory bacteria like Haemophilus, Klebsiella, and Streptococcus. And then we saw a decrease in Porphyromonas, Actinobacteria, and Gemella. Now, in non-type 2 asthma, we saw an increase in definite inflammatory groups of bacteria like Fusobacterium and even Porphyromonas. But then we interestingly saw a decrease in certain families like the Lactobacilli family and the Lactobacilli order, which is often associated with a healthier gut microbiome and a healthy lung microbiome as well. Now let's discuss the connection between the gut microbiome and our endocrine system. This study in JAMA looked at the association between insulin resistance and type 2 diabetes with gut microbial diversity. And what these researchers found is that type 2 diabetics are more likely to have gut dysbiosis. The gut microbiome not only helps regulate our hunger and satiety hormones, but it can also impact insulin release and sensitivity. When researchers have looked at the stool samples of patients with type 2 diabetes, they found that they had fewer levels of positive and health-promoting bacteria like Lactobacillus fermentum, Lactobacillus plantarum, Rosbergia intestinalis, and Akkermansia. Now, these groups of bacteria are important because they have been shown to improve glucose metabolism and insulin sensitivity while suppressing pro-inflammatory cytokines that tend to be present and overexpressed in the presence of lipopolysaccharide. Now, interestingly enough, researchers have found that metformin, which is the first-line pharmacotherapy for type 2 diabetes, has been shown to act primarily in the gut and alters the composition of the gut microbiota. Metformin has been shown to increase butyrate production and increase propionate production, two very important short-chain fatty acids. And it's also been shown to increase levels of Akkermansia muciniphila, which is our mucin-loving and mucus layer rebuilding keystone species that is critically important for overall health, especially metabolic health as well. Researchers have dug a bit deeper on the relationship between the gut microbiome and metformin. When they looked at the microbial mapping of patients who were type 2 diabetics, they found that at the phylum level, these patients had fewer firmicutes or firmicutes than the control group. They had more proteobacteria than the control group, and they had fewer amounts of butyrate producing bacteria like Roseburia. What researchers have found is that the abundance of that gram-negative bacteria that have lipopolysaccharide can stimulate the immune system and stimulate toll-like receptors, which further drive the inflammatory process. Type 2 diabetes patients often also had low alpha diversity, so they had fewer species of bacteria as a whole in their microbiome. The gut is also considered a target organ for our thyroid hormones. That includes T4 or thyroxine and T3 or triiodothyronine. Intestinal epithelial development has been found to be controlled by thyroid hormone signaling. In fact, thyroid hormone receptors are expressed on the surface of our intestinal epithelial cells. Disruption in the microbial composition in the gut has been associated with Hashimoto's thyroiditis, Graves' disease, and Graves' orbitopathy. There's another connection between thyroid health and the gut microbiome, and specifically gut dysbiosis. We found that there is an enzyme called intestinal alkaline phosphatase, or IAP. This is a brush border enzyme that's primarily expressed on the duodenum in the small intestine. Now, IAP is very important because it works to dephosphorylate or deactivate lipopolysaccharide, and that prevents it from translocating across the colonocytes, across those weakened tight junctions, and into circulation. Now, what's important to note is that IAP, the gene that codes for IAP, is a T3 responsive gene that directly links its expression to thyroid hormone levels. That suggests that patients with low thyroid production or hypothyroidism may be more likely to produce less of this intestinal alkaline phosphatase enzyme, which makes them less able to detoxify lipopolysaccharide. Studies on animal models discovered that decreased IAP gene transcription was found in hypothyroid rats. And when they knocked out IAP in zebrafish, those animals became more susceptible to LPS toxicity. There are a number of microbial patterns in autoimmune thyroid disease. Let's look at some of the bacterial strains that are associated with increased or decreased expression of different antibodies associated with autoimmune thyroid disease. We see here on the left that the bacterial strains associated with a decreased expression in TPO and thyroid receptor antibodies include Bacteroides, the Dorea species, and Faecalibacterium prausnitzii. We also see that certain bacterial strains have been associated with an increased expression of TPO antibodies and thyroid receptor antibodies. Now, it's important to note that this is likely due to molecular mimicry. So for whatever reason, the structures on the surfaces of these bacteria mimic the structure and surface of TPO and thyroid receptor. Now, these bacterial strains species include the Blausch's species, Lactobacillus, Ellis types, and Ruminococcus. Now, the three bacterial strains that have been seen in patients with Hashimoto's thyroiditis include Lachnoclostridium, Bilophila wadsworthia, and Klebsiella pneumoniae. Let's talk about dysbiosis and the immune system. Dysbiosis has been associated with many immune-related human diseases, but in many cases, it remains to be established whether dysbiosis is a cause or consequence of the disease. So let's look into what the research has highlighted as to delineate the connection between gut dysbiosis and immune dysfunction. This study, published in 2020, highlighted the crosstalk between the gut microbiome and the innate immunity. Specifically, it also highlighted its implication in the development of autoimmune diseases. What they found in this study is that dysbiosis of the gut microbiota has been shown to be related to a variety of alterations in the immune system. Gut commensal bacteria are known to have many vital roles in the establishment of a regular innate immune system. So in turn, dysbiosis of the gut microbiota can cause an alteration in innate immune system function, structure, and support. Various studies revealed that the structural buildup of the GALT, or the gut-associated lymphoid tissue, relies on the gut microbiota. The formation of gut secondary lymphoid organs like Peyer's patches greatly depends on lymphoid tissue inducer cells, and those cells have crosstalk between the gut microbiota and the function and development of the immune system. Researchers have found that microbiota colonization in the GI tract during early life affects the development of different T cell populations. It will affect the development of T helper cells, including T helper 1, T helper 2, T helper 17, or into regulatory T cells. Now, regulatory T cells are particularly important because these are considered our immune fire extinguishers. They cool off the inflammatory process that is initiated by T helper 1, T helper 2, and T helper 17 cells. Lastly, let's discuss the gut microbiome and the genitourinary axis. In many ways, the vaginal microbiome is the opposite of the gut microbiome. A healthy gut microbiome is rich in diversity and may not contain a lot of lactobacillus species, but the vaginal microbiome is low in diversity and is predominantly lactobacillus. Lactobacillus lowers the pH through the production of lactate, which helps keep the vaginal pH at a healthy lower level. Now, a healthy vaginal pH should be anywhere between 3.8 and 4.5. This pH range helps facilitate the abundance and growth of 4 of the most dominant lactobacillus species found in the vaginal microbiome. They include Lactobacillus crispatus, Lactobacillus gasseri, Lactobacillus jensenii, and Lactobacillus iners. Now, it's important to note that Lactobacillus iners may be also associated with vaginal dysbiosis. As I mentioned, Lactobacillus is critical for healthy vaginal microbiome health. The presence of Lactobacillus species lowers pathogen abundance because Lactobacillus species can produce lactate, bacteriocins, and hydrogen peroxide. Bacteriocins are toxins produced by bacteria to inhibit the growth of similar bacterial strains. In fact, a low abundance of Lactobacillus species allows for an increase in opportunistic and pathogenic bacteria. That includes Gardnerella vaginalis, which is the primary culprit behind bacterial vaginosis, along with Prevotella bivia, Atopium vaginae, and Mycoplasma hominis. Lactobacillus species can inhibit the adherence of pathogens by producing biosurfactants that compete with the attachment sites. Or Lactobacillus can co-aggregate with the pathogen and allows the pathogen to be swept away by the host body fluid. The vaginal microbiome can be impacted by age, hormones, sexual habits, medications, the use of certain vaginal products, diet, stress, tobacco use, and of course, the gut microbiome. The gut microbiome does play a role in the development and health of the vaginal microbiome, and that's due to the gut and vaginal microbiome crosstalk. Of course, this is partially due to the close proximity of the vagina to the anus, which allows translocation of certain species after being shed in feces. It's also due to the close proximity of the urethra to the vagina, and the vaginal microbiome can also play a role in the development of urinary tract infections. There is a connection between the gut microbiome and the vaginal microbiome. Researchers have found that women with BV-associated bacteria in their stool reported a higher incidence of bacterial vaginosis infections. Those bacteria include Gardnerella vaginalis, Prevotella bivia, Atopobium vaginae, and Mycoplasma hominis. Now, gut probiotics containing lactic acid producing bacteria like Bacillus spores can help maintain the vaginal pH and obstruct the growth of pathogenic bacteria in the gut. By inhibiting the growth of unfriendly bacteria like E. coli, gut probiotics like Bacillus spores can help promote vaginal microbiome health by supporting healthy gut microbiome health. Women with a higher concentration of Lactobacillus crispatus in their stool were more likely to have healthy vaginal microbial composition. Now, dysbiosis of the urinary microbiome may directly be associated with disorders of the urinary tract, like urinary tract infections, interstitial cystitis, and urinary incontinence. Here we see that the bladder axis crosstalk does involve the gut and the bladder. A healthy bladder is usually comprised of Lactobacillus species. Overactive bladder or urinary incontinent tend to have a bladder that contains Atopobium, Gardnerella, Proteus, and even Snethia. Recurrent UTIs tend to occur in a bladder that contains E. coli, Gardnerella, Pseudomonas, Sphingomonas, Streptococcus, and many others. Interstitial cystitis bladders tend to be colonized by Brevibacterium, Citrobacter, Lactobacillus gasseri, and Serratia. In a healthy state, the bladder and urinary microbiome usually contains mostly Lactobacillus, followed by Prevotella, Corynebacterium, Gardnerella, and even a bit of Shigella. The pH of the bladder should be somewhere between 4.6 and 7. However, exposure to different risk factors like indwelling devices like catheters, age, low estrogen levels, barrier contraceptives, vaginal infections, and even existing urinary conditions can lead to UTIs. Spinal cord injuries, antibiotics, and even conditions like diabetes can also increase someone's risk of developing a UTI. When someone has a urinary tract infection, the pH of the bladder increases to anywhere between 7.8 and 9. This basic level of pH encourages the growth of pathogens like E. coli, Enterococcus, Staphylococcus, Gardnerella, and Lactobacillus. Thank you so much for joining me today. In summary, dysbiosis is an imbalance between beneficial and harmful bacteria. This microbial imbalance impacts all areas of our health. Gut dysbiosis has been associated with anxiety, depression, Alzheimer's and Parkinson's disease, asthma, type 2 diabetes, autoimmune diseases, and urinary tract infections. Restoring gut homeostasis is critical for supporting overall health and reducing chronic disease. Thank you so much for your attention, and I hope you have a wonderful day.